As a derivatives of this metabolites or the natural product, now small molecules are very important the tools for chemical biology the study. And these small molecules can be the potential drug candidates too. And to use this small molecule for chemical biology study, the first important the point is the molecule should be smoothly introduced to biological system. And this property is so-called drug-like the property. So by looking at these two molecules, can you tell which molecule can enter the biological system more smoothly than the other? So in other words, which molecule is more drug-like? If you think about those entry of the molecules in the biological system, the first barrier is the lipid membrane, lipid bilayer membrane. And there was one medicinal chemist named Lipinski. He compared thousands of commercial the drugs and tried to extract out the important physical property of those molecules which can determine those absorption, whether poor or other good. And he found some rules. Those rules we call Lipinski rule, or there's many 5 has been used there. So sometimes this rule is called as the rule of 5. And let's see what kind of rule he found. First of all, he found if the molecule's molecular weight is above 500, usually they have poor absorption. They cannot penetrate the membrane. Interesting, isn't it? So this is the first time now we can start to say good small molecule or drug-like small molecules, the molecular weight, the limit is 500. So if the molecule is smaller than 500, we call them small molecule. If it's bigger than that, maybe we said metabolite can go up approximately 1,500 molecular weight. Then between 500 to that limit, 1,500, we call them as kind of a big small molecule or big metabolite, relatively. So when we call drug-like small molecule, molecular weight is less than 500. Okay. And partition coefficient, so the log p value. So if you calculate log p, we call C log p. If it's this number is high, then that means the molecule is hydrophobic. If the number is low, then the molecule becomes hydrophilic. If log p value is above 5, they cannot enter the cells because it's too hydrophobic. They just encounter any kind of a membrane structure, they're just stuck to the membrane. They cannot penetrate. If log p value is lower than 2, it's too hydrophilic. That's why it cannot penetrate the membrane. They just stay outside of those membrane. Lipid bilayer membrane, it has lipophilic area inside and hydrophilic area outside. So the hydrophilic molecule, they can interact only the surface of membrane, but they don't have a chance to penetrate that membrane structure. So when the log p value is between 2 and 5, they have a reasonable chance they can penetrate the membrane and can get inside the cell or coming out of the cells. So this is partition coefficient, the rule. And if hydrogen bonding donor number is above 5, this molecule has a poor absorptions. That's what we found. And also, hydrogen bonding acceptor number becomes bigger than 10. Now that molecule also has a problem for the absorption. So these two, 5 and 10, for the hydrogen bonding donor and acceptor, we are going to talk about this a little more later. And this rule has been published originally the 1997 in Advanced Drug Delivery Review. And in this review, the journal, this paper has been the most cited paper. And I just checked today, as of August 21, 2016, this single review paper has been cited more than 10,000 times. If one paper has been cited more than 10,000 times, it may have a quite good chance to get the Nobel Prize. And interestingly, this paper is so popular and has been allowed to make revisions. So it seems like the multi versions of revision of this original paper has been updated and further. In addition to this original, the rule of 5, now there's another, the rule is added to complement those properties of absorption. If molecules, the rotational bond number is above 10, this molecule also has poor absorptions. Okay, let's take this molecule as an example. Can you analyze this molecule's the property following Lipinski rule? Then you need to figure out the molecular weight and all other parameters, right? Now these days, if you look at, if you use the ChemDraw, then you can extract out this information easily. Now you can see here chemical formula and the molecular weight. So if you look at those molecular weight from ChemDraw, you can get the two numbers. So one is the exact mass and the other one is the molecular weight. We are going to talk about it later again. Now this molecule, molecular weight wise, that's 272. It's less than 500. Okay, it's not problem for the molecular weight of the parameter. And even you can get C log P value from ChemDraw. This molecule, my version of ChemDraw, I could get 2.1. So C log P is also between 2 and 5. Maybe absorption may not be a problem for this molecule. Now here, can we count those hydrogen bonding donor? To make those analysis simple, he just put the nominal, the hydrogen bonding donor. So he counted hydrogens directly attached to oxygen or nitrogen. That's how he counted hydrogen bonding donor. Can you find how many hydrogen bonding donor here? Yeah, there's only one hydrogen here attached to the oxygen, or we can call this is a proton. So this proton here, none of other the hydrogens. So it has only one hydrogen bonding donor here. How many hydrogen bond accepted here? Also, to simplify those rules, he just counted only nitrogen and oxygens. Okay, now let's count. How many here? Here, 1, 2, 3. Do you count this one out? We do count. So this one acts as a hydrogen bonding donor, but at the same time, it can act as a hydrogen bonding acceptor. That's why 1, 2, 3, 4. So it's also less than 10. Hydrogen bonding donor is less than 5. And how about the rotational bond here? How many rotational bonds can you find? 1 here, 2, 3, 4, 5, and 6. How about this H and O? It is only one atom here, so we don't see the bulky volume effect here, so we don't count this. So then, this is less than 10 rotational bond. So this molecule seems like, at least by the physical property wise, this molecule we judge drug-like molecules. Now you may ask this the parameter log P. What is log P? Log P is, the P is a partition coefficient, and we just put log values for this. This is partition in between two solvents. Those two solvents are normal octanol and water. And you mix it together one molecule in this two-phase system, and then measure those partition on octanol over waters. That's why if this log P value is 0, that means the distribution of the molecule in these two solvents is 1 to 1. So if log P value is 1, that means it's approximately 10 to 1. If it's 2, then that's 100 to 1. Then log 5 is 100,000 versus 1. Octanol is a hydrophobic solvent. That's why the higher number of the log P value means the molecule is more and more hydrophobic. Now see here the calculation. So to all of these informations you can get from ChemDraw. Okay, let's look at a little at the macro views. So there was a study to look at what is the main reason for the failure of drug development. And then if you look at this, the pie chart, which one is the biggest one? So biggest one here on the right side, 39% of those drug candidates, they are failed because of the problem of ADME. So then naturally we are curious, what is ADME, right? ADME is a pharmacokinetic parameter. And what is a pharmacokinetic then? Pharmacokinetics means it's how our body handles drug molecules. When I explained the Lipinski rule, I just simplified our body as a one cell, and we have a membrane outside, and then whether the drug molecule can get inside, crossing those membrane structure or not, that's kind of a simplified view. But actually our body is much more complicated. So to take those compound or the drug molecules into our body, the first step should be absorption. That's represented by A. And then distribution. And the molecule or the drug candidate should reach its target or the portion in the body. And then metabolized. And then eventually secreted. So this whole process we call ADME. Then the activity of the molecule we can call, in comparison to the pharmacokinetic, that kind of a potency we call pharmacodynamic property. Okay, so we have two different aspects. Pharmacokinetics is how our body treats the molecules, how to absorb it, and how to distribute, and how to change the molecule, and eventually how to remove the molecule. Pharmacodynamic parameter in terms of potency or efficacy, that is how the molecule is effectively the affect our body, and then generate side effect or the good effect. Those parameters are the pharmacodynamic parameter. Of course, those are very important, but if the ADME, the pharmacokinetic parameter is very bad, then the molecule does not have a chance to do good things for our body. If you look at this, the ADME part, so the first part should be absorption. It can be orally or by injection introduced into our body. And then when it is transferred to the heart, and then it can be distributed the whole of our body through the blood-vessel system. And then when it passes through liver, and then it is metabolized by different other enzymes. And eventually it is removed from our body by the excretion of the pathways. Then we need to think which part of the body is really inside or outside. I just briefly mentioned this in the first lecture, but we didn't have a chance to discuss. So this part we are going to discuss in the next lecture. Okay, now let's move to pharmacodynamic part. What information can you get out of this data? First of all, by looking at those concentrations, seems like the concentration is increasing from left side to right side. And unit-wise, this is a log value. And log 0 is 1, right? And then unit is micromolar. So this is 1 micromolar concentration. And 1 should be 1 and 2 around here. 1 is 10 micromolar, 100 micromolar, right? Let's go down here. Minus 1, minus 2, minus 3. Minus 1 is what's concentration? It should be 0.1 micromolar. That means 100 nanomolar. And then 10 nanomolar and 1 nanomolar. And this unit is called as IC50. IC50 is inhibitory concentration 50. So that's the opposite of the activity, but activity can be defined either activation or inhibition. If you put very small amount here, this is 100% of activity. And then if you add a large amount of those drugs, it can be fully inhibited here. Your window is, this is 100% inhibition, this is 0% inhibition. If you talk about this as the efficiency, then this is the opposite. This is 100% of the effective, and this is 0% effective. In this case, this is measured by product formed. If it is a product, maybe the target may be enzyme, which is catalyzing some other chemical reactions. In this case, this is a MMP, it's a protease. Now here, if I put A, B, C, 3 molecules, which molecule is most active molecule? Can you tell? From this graph, if you look at the IC50, similar to the LD50, LD50 case, it was a lethal dose 50. C and D is just a different unit. D is a dose. It's usually milligram per kilogram kind of unit. Concentration, C, is usually expressed by molar concentrations. If this is 0%, this is 100%, IC50 value should be defined here by putting around the half line here, and then IC50 value should come from this number, and then B should come here, C should come here. Then we can tell number-wise IC50, C has the highest number, A has the lowest number. So IC50 value-wise, the lower the stronger or the higher the stronger? Yes, the lower the stronger, right? Even though you put a small amount like this, still you can get 50% inhibition of those enzymes. So that's how we read this data. Pharmacodynamic parameters can be either positive or negative directions. If it is activating, we call those kind of molecule agonist. If it's inhibiting, those are antagonist. So in this graph, can you tell this is agonist or antagonist? Previous IC50 value case, that was the inhibitors. Inhibitors we can call that's antagonist, right? Now in these two graphs, cyclic AMP and arrestin, are these agonist or antagonist? Actually, there is a hint on the bottom. Both of them has agonist. The point here is, depending on how you define your y-axis, this is percent inhibition. Percent inhibition is a high, that means no inhibition here, so that's the activated state. Percent inhibition is going down, that's a fully activated here, so this is activation graph. Now directly you can put maybe percent activation. Now originally it was low, but it's going to the high level. So this is a positive way to draw those are the agonist effect. So don't be deceived by these directions, whether the graph is going down or going up. Okay. Now here we need to discuss about potency and efficacy. The first parameter we call potency. These two molecules has almost the same 100% the activity. Then in this case, we can define ED50, that is effective dose 50. This should be around 100%. Then ED50 wise, that should be defined around the half effect here, right? Then if you call this is A and B, which molecule has higher potency? Yeah, it's A, because A should have lower number of ED50 than B, so that means potency can be defined by the amount required to get same amount of the activity. That's a potency. That's why using small amount of A, you can get same effect of B. That's why we call A, we can say A has higher potency than B. But sometimes the maximum activity is not different among two molecules. So this molecule can go even higher here. So in this case, A and B again. A molecule can reach up to this much of effect. B can go only this much. Then also the effect is almost saturated. Even though we add a lot more of B, it cannot reach up to here. This difference we call efficacy. So A molecule has higher maximum, higher potential maximum, and this molecule has higher efficacy than B, we call. If A has 100% efficacy and B has only 60% of efficacy, maybe you can say B is partial agonist for A. The real situations can be more complicated. Now in this case, molecule N and molecule B, which one has a higher potency? Then we compare those ED50 values, right? Then A has ED50 value here, B has ED50 value here. So A is more potent than B. But how about efficacy? Which molecule has a higher efficacy? That is B. B can go up to here if you add more amount, but A is already saturated here, it cannot go higher. So B molecule in this case, this is the higher efficacy molecule than A. How do you compare these two molecules? MDX1401 and MDX060. To make it simple, let's just say blue pill and red pill. Which compound has a higher potency and which compound has higher efficacy? Did you get it? Well, in this case, blue compound has enhanced efficacy and also higher potency. Okay, so far here, I tried to introduce some basic terms we are going to use and over in description of drug activity and also biological functions. So we heard LD50, ED50, or IC50. Those D and C can be easily interconvertible, as said. So either dose or concentration. So milligram per kilogram or the molar concentration, it can be any time, can be interconvertible. Okay. And also, the potency and efficacy, it can be used for agonist for activation measurement, or it can be used for inhibition measurement too. So if you develop enzyme inhibitor as a drug candidate, then higher potency means it has a better inhibition activity, right? So every case is, if you define your y-axis properly, even same agonist or antagonist, it can go up or down depending on how you define your y-axis. So that's exactly the same way potency or efficacy also can be used both for agonist and antagonist. Okay, that's why whenever you encounter some confusing other situations, the best way is you start with your definition first, whether you are describing inhibition or activation. And based on that, if you draw and you provide your answer, then you are safe. Okay? In your study, if you knew your target enzyme, then maybe the mechanism is straightforward. In the case, maybe more practical problem is whether your compound or your natural product attacks only one enzyme or other enzyme too as a side effect, then maybe the more major concern you need to figure out. But if your natural product or small molecule or the tool is totally new material, nobody has made that kind of material, you are the first one in the world, you found very interesting material, then how can you figure out the mechanism of the molecule? If possible, by just one unified test, not testing too many things. Do you have a good idea? One possible solution was provided by the National Cancer Institute in US. NCI has been collecting many compounds from all over the world and then testing those compounds as kind of academic services. In early stages, they tested directly the compounds in mouse. And those mouse has transplanted the tumor and check whether it can cure the tumor or it kills the mice as a side effect. But later stage, they changed this screening into 60 different human cancer cell lines. So this is so-called NCI 60 cancer cell lines. Over the time, those cancer cell line has been changed a little too, but this has been used as a standard cancer cell line test for many compounds. Almost million compounds, maybe above million compounds, has been tested in the same panel or similar panel. Now, that accumulated data can provide those patterns. How those compounds, even new compounds, across these 60 standard cancer cell lines, if you compare those cancer cell line response as a pattern, maybe it can provide the insight of mechanism of any compound, any new compound too. Okay. Using that idea, let's say they measure only 3 data points. So this is the raw data they are generating. You can see here 60 different cancer cell lines survival the curve. Now again, you can see here molar concentration and log value, and here what is the concentration here? 10 to the minus 6 molar, that's micromolar, right? 10 micromolar, 100 micromolar, 100 nanomolar, 10 nanomolar. So that means, seems like the data looks like 1, 2, 3, 4, 5 concentration tests against 60 different cancer cell lines that they keep as a standard cells. From this data, this is the starting point of cell numbers. This is the growth of those cells. And if cell number goes below this line, that means cells are dying. If you had a very small amount here, now this is the starting point, cells are actually growing, right? And then this is cells are dying to this direction if you had more amount. They pick up 3 data points. First one, here, growth, this 100% growth, then this is 50% growth, this concentration. That's what they call GI50, growth inhibition 50. Okay. Now this one, 0 value here, this can tell you whether cells can grow or not. If they stay here, the compound has growth suppression, but it is not killing the cells. This is what they call TGI. It's a threshold growth inhibition. Now, half of those cells died from comparing to the starting of the cell line. Now this one is really killing the concentrations. This is called as LC50, lethal concentration 50. They collect 3 parameters like this. Using these 3 data points, so GI50 and TGI and also LC50. Now this data point, you can put mean values among those 60 cancer cell lines. And some cancer cell line shows more sensitive to this compound. Some cancer cell line shows more resistant if it goes this way. Then by putting those data, now this is one compound, the Taxol. Taxol has this kind of a profile. These cancer cell lines, this is 60 different cancer cell lines. And this cancer cell line is more sensitive to the Taxol. And this one is more resistant. By using this kind of pattern analysis, now if they have a similar pattern, even though the concentration can be different, but relative values, relative sensitivity to this compound among those cancer cell lines can be similar to each other, then let's say even though this compound is a new compound, if enough database has this kind of a pattern, by doing this pattern analysis, we can figure out, okay, this compound may have similar mechanism with the Taxol. Taxol is an anti-tubulin compound, then this compound may be anti-tubulin compound too. Using similar analysis, by analyzing thousands of tens of thousands of compounds analysis, now some of those classes can be classified. Okay, this pattern of those other cell, then you can tell this is a topoisomerase 2 as potential the targets. Quite interesting approach utilizing huge data. These days more popular word maybe big data. Using different kind of big data. In a sense, this is kind of a small big data or small scale big data. But using this kind of a pattern analysis, even without knowing exact molecular target, at least we can get out some clues of those compounds mechanisms. More extended the approach for this. NCI compare the approach, they utilized 60 cancer cell line, 3 data point. So those were GI50, TGI, and LC50, 3 concentration point they used as an input. Now we are getting into the omics field. And then one of those omics is the genomics. And different cells has a different genomic profiling, that's a transcriptome analysis using DNA microarray or sequencing itself. And similar method can be applied to drug the mechanism of the study. If the same cell is treated with 2 different compounds, so let's just say it can be either human cell or mouse cell, or it can be just the cultured cells like NCI's cancer cells. By putting different compound or different the drug candidates, we can measure those changes of those cells in terms of mRNA expression level changes. Now here, transcriptome, how many transcriptome we have? Genome number was about like more than 20,000. Transcriptome maybe 100,000. Depending on those chip design, we can say now 100,000 of different output we can measure, and some of them is increased, some of them is decreased. And by analyzing those patterns, then we can classify those compounds to compounds effect using this really big data. Then it can give us very good clues that these two compounds are in the similar the mechanism. This compound is quite different from this mechanism. Kind of approach is possible. In this case, to connect even different data set, connect to this into common the idea approach wise, we call this connectivity map the approach. So after this, I will show you the original, the scientist, the description of this technology.
My name is Todd Golub.
And I'm Justin Lamb. With our collaborators here at the Broad, we've developed what we call the connectivity map.
What we're trying to do with the connectivity map is to apply the power of genomics in a way that could make significant impact on the way we discover drugs. That process historically has been a very ad hoc one, where there haven't been ways to systematically figure out what it is that a particular drug actually does inside a cell, or figuring out ways to determine for a particular disease what drugs are there that might best treat it.
The basic idea of the connectivity map is to say, okay, these are two domains, meaning disease and chemistry, that look very, very different. The people who participate in them are very, very different. You know, there's physicians on this side and those hard-nosed organic chemists on this side. They can't interact because they speak different languages.
And so the idea of the connectivity map is to find a common language that both biologists and chemists can use and understand, and use this common language to establish connections that we didn't previously know about. The language we chose is what's called the gene expression profile of a cell.
Every cell in your body has the same set of genes, about 20 to 25,000 of them. The reason you've got many very different types of cell is that each cell type turns on or expresses a different set of those genes. This cell expresses this handful of genes at these levels, and this one expresses a different handful of genes at different levels. I shouldn't say handful, right? Because that sounds like a low number. But you know, this bucket of genes and this bucket of genes are expressed at different levels.
Just as different genes are turned on and off in different cell types, so different genes are expressed in disease cells than in normal cells. And that pattern of which genes are turned on or off is the gene expression signature of that disease. Some of the machines we use to read a cell's gene expression signature are on display in this exhibit. You can listen to Stacey Gabriel describe how the signatures emerge from the machines in the form of an extremely complex two-dimensional barcode.
The analogy with barcodes, you know, on your groceries is a good one. You go and you buy, I don't know, coffee or something, and there's a barcode on there. You've got no idea what that barcode is. By looking at the barcode, you couldn't tell it was coffee. But if you go to the checkout and the guy scans it, he's got some way of interpreting those lines into coffee, and it's $11.99 if you buy the good stuff or something, right? So you know, the gene expression profile, although it probably explains the disease in some way we can't interpret, it's a barcode for the disease.
Well, just as a disease has a genomic barcode, so does a drug. If you take a cell in the laboratory and treat it with a drug, some genes stay the same, some genes go up, and some genes go down. And so if we can describe diseases and drug action in the same kind of language, the language of which genes get turned on and off, now all of a sudden we can see connections between diseases and drugs because we can see the similarity in these signatures.
The genius of the connectivity map is that there is no genius, right? It's actually very, very simple. And in many ways, by making dumb choices, making executive decisions, just saying, okay, this is not really that hard, let's do something simple, it turns out to work well. We've already profiled many of the 1,314 drugs approved by the FDA. One of the things we hope to do with the connectivity map is find new uses for some of those existing drugs.
In fact, we're already discovering that there are drugs that induce multiple different signatures in a cell. Maybe the drug was developed with a particular signature in mind, but now we're finding that drugs that have been available at the drugstore for years are activating signatures we never knew about, suggesting that these drugs may have new uses.
A nice example comes from a collaboration with Scott Armstrong, a pediatric oncologist at the Dana-Farber Cancer Institute. He's spent a good portion of his career trying to figure out why about half of his patients with acute lymphoblastic leukemia are essentially cured by a drug called dexamethasone, while the other half of his patients, he can give them this drug and nothing happens. The drug doesn't work.
And so it occurred to us that we could make a signature of dexamethasone sensitivity by comparing the tumor cells from patients that are sensitive to the tumor cells from patients that are resistant. So we got a list of genes that represent the dexamethasone sensitivity signature. And then we reasoned, well, if we could find some other drug that induced a cell to turn on that signature of dexamethasone sensitivity, maybe it would actually make the cell sensitive. And so we took that signature and compared it to all the signatures in the connectivity map database and said, does anything look like this signature? And the map said, yes, there's one drug, a known drug, that's already FDA approved and used in the clinic for something else called sirolimus, also known as rapamycin.
So Scott went back to the lab where he has all the reagents to do these experiments very, very quickly, and showed that indeed, if you treated dexamethasone-resistant cells with dexamethasone plus rapamycin, they would now be killed to a degree greater than had ever been seen before. And because rapamycin is already FDA approved, it was immediately obvious to suggest that you could use that combination clinically. And he's now enrolling patients in a clinical trial to test that novel combination.
At the same time as we were working our way through the FDA-approved drugs, we're also looking at chemicals that are being created for the first time being synthesized at the Broad. And those are very interesting because these are molecules that no one's ever seen before. It's not obvious to know how to actually figure out what they do. One way to figure this out is to make a signature of them. I'd hope that in 5 years, say, the database is 100 times more data than it does now, and that interacting with the connectivity map becomes a routine part of doing biomedical research.
Todd's likened the connectivity map to Google. It's actually very much the same. But rather than type in a word or a phrase in that little box, you essentially type in a list of genes differentially expressed in a piece of biology that you care about. You click one button, and now you're presented with a rank-ordered list of chemicals ranked by their similarity to the list of genes that you uploaded in the first place. So I think the analogy is quite strong. And also because it's free, it's quick, it happens in real time, and you can do it from anywhere. And it's good fun to do, right?